So research, I think it's kind of there's a little bit of mystic unknown to research. A lot of you will end up doing research projects as your student here. Um, it sounds fancy, and it can be kind of fancy because a lot of times research involves big money. You have to get grants and propose money sources and all this. But it's really the kind of thing that anybody could do, but it's basically the foundation of science and how we find answers. So in a class, I think the information is often presented to you like it's truth. It's already done. We figured this out. This is how it works. But all of the features, the topics that we present in these science classes, these are based on evidence that has been presented previously, sometimes questions that weren't all solved at the time they were asked, right? So we say a lot of things very certainly about how DNA works, for example, but there was a time when we didn't have any idea what DNA was or how it worked. Same thing for plants. So what I have here on this slide are some like definitive statements. Caterpillars induce jasminates, that's a form of chemical signal, jasminates in flowers, and alter plant responses to a second attacker. So caterpillars chewing on a plant cause the plant to defend itself against other herbivores. Done deal, we know that. Plants roots use sound to locate water. Leaf shape deters plant processing by a weevil. Does it make you want to know more? Maybe? Hopefully. All right, so all these conclusions, kind of working backwards, they're coming back from questions. So before there was a conclusion, there was a question. Are plants able to respond and defend themselves when an animal is chewing on them? So I like the occasional Calvin and Hobbes cartoon. This is basically hypothesis testing. What will happen if I jump on this board? What does it mean to have a hypothesis? What's that? A question? All right. I think there's a little more to it. Uh-huh. Hypothesis is a little more like the answer to the question, right? Okay. Yeah. So a lot of people, I think they get trained on this if-then for hypothesis. Yes. I guess maybe we're using the word answer a little differently, right? So you can have like a hypothetical answer. I think this is going to happen. A prediction. Is that better? All right. So we definitely have questions. The if-then statement. So if I jump on this board, I think the giant snowball will fall on my head. The if-then is kind of based upon doing a certain kind of experiment, and that's an important part of science and can be one of the more difficult things to line up. Like, how do you design the right experiment to get your answer? Right. What do you think the answer to the question will be? Right. So we have a question, an experiment. The experiment we'll talk a little bit about basically lining up a way that you can discern one of two outcomes. So I think this is going to happen. Maybe two possible outcomes, right? Jump on the board and this snowball will rocket into space, or it'll fall back on my head. Right, so hypothesis, yeah, basically an informed prediction about how usually a specific experiment will turn out. And as we'll talk today, it's important to have a rather narrow scope for every one of your experiments. Probably should have more than a Calvin and Hobbes cartoon here. So if you have a hypothesis, all right, let's do a simpler experiment. If I add fertilizer to a plant, there's our if statement. What happens if you add fertilizer to a plant? Okay. If I add fertilizer, the plant will grow better. Than if I hadn't added fertilizer. Uh, right. So hypothesis here. A lot of times the hypothesis is kind of what you want to happen. I think we can be objective researchers and still have kind of a preferred outcome. So let's say you read your literature and you find out that in lots of other plants that happens if you add fertilizer, they grow better. You think in your specific example, you've got a plant and you add fertilizer to it. You think that will be the same effect. So a lot of times this is called the, I don't know if I like any of the language on this, but basically what we usually think about for hypotheses is what's often called the alternative hypothesis, a research hypothesis or an experimental hypothesis. And anytime you have one of those hypotheses, you also have a null hypothesis. So I mentioned earlier that you end up competing between two possible outcomes. So at the end of your experiment, you should say either I'm going to side with this hypothesis or the other hypothesis. It comes down to one thing or the other. Have you proved it? Have you not proved it? So our null hypothesis here, and I'm using some H sub 0 or H sub naught, sometimes they call it, is the null hypothesis. And it's kind of like your fallback hypothesis. So if you don't convincingly demonstrate that your experimental hypothesis is true, and you fall back on this one, which is more or less the opposite of the first one. So the plant, if I add fertilizer, the null hypothesis is that the plant will grow no better than if I hadn't added fertilizer. All right, so a couple chats from Tom in here. Our hypothesis that the plant will grow better with fertilizer, definitely true, depends on the plant and the fertilizer. There are plants that are very sensitive to nutrients and you can kill them by adding extra nutrients, or maybe your particular plant, that's the one thing that it doesn't need right now. And I guess maybe related to that, his second question, what is the hypothesis that says the fertilizer will outright kill the plant? I don't know. Anybody know? I guess there's a whole wealth of scientific don't end up being published, and I think that would probably fall into that category. So if you were supposing that fertilizer would make your plant grow better and you ended up killing your plant, you probably don't. If you were aiming to make a plant grow better, you probably would not publish or even try to publish the results in that way. You could, I don't know. Equally, all right, so good questions. So I guess, I've got this alternative hypothesis called H1. Sometimes people number these as H1, H2. You could, at the same time, test if the plant grows worse than if you hadn't added fertilizer. So yeah, I guess I build this as one of two possibilities. You could have more than two possibilities. What I'll end up presenting today are experiments and data that really have you choosing between two possibilities. So I guess you could do a test. You could get your data and at the same time find out if growth was better with fertilizer, and you could also at the same time find out if it's growing worse with fertilizer. So all right, all of these are important. As far as scientific knowledge, I think the connection of nutrients and growth, maybe that's not as well. I guess I don't read the journals that would figure that out, but every plant across the world has some level of tolerances. Nutrients, water, sunlight, soil minerals, CO2 availability. So you could take a plant and run it through all kinds of trials and find out what conditions cause it to grow best. A lot of people do this unofficially. Let's say you're just running a greenhouse and you want to make your plants grow better. You might not be collecting data all the time, but figuring out over time what makes your species grow better. All right. I guess I'll leave that as it is. So I guess before I go back to the slides, how do you know if your plant has grown better? What's that? Okay, control. Right. Right. So a lot of our experiments have a control and a treatment. Right. So we could run an experiment where we have control and a treatment. You can't just add fertilizer to a plant, say, looks like it grew better, and call it a day. So a good experiment has a reference point, a control. So I have two plants of the same kind. I'm going to add fertilizer to one and not add fertilizer to the other, and keep everything else the same. So if you are able to, the best way to do this is in a controlled environment, controlled amount of light, temperature, water. And if there is a difference between control and treatment, you would see that even though everything else was the same. Tom also chimed in something I was trying to get to, that you need, or you prefer to convert this into something numeric. So what does it mean to grow better? So height, did it grow taller? Biomass, does it weigh more? Did it put more mass into the plant? Right, so choose something that matters to measure, sets you up to do the numerical comparison, which is something we'll definitely emphasize today. All right, so far? Seems pretty simple so far. Okay, so we already spelled out experiments and treatment and control, sorry. So an experiment is a situation that you set up or you manage to observe that boils the difference down to as few variables as possible. So you can really observe that difference. So if we go back to the fertilizer example, if you grew your fertilized plants in the sun and your not fertilized plants in the shade and you try to compare them, you're not doing a very good experiment. You want to just make it so you're comparing one with the other. Kind of enjoy the stock photos I found for experiment. They just had a series of kids that they put in the same setup and make them look like they blew something up. It's science, right? Blowing stuff up. Okay, so you can have your hypothesis, but to really be able to demonstrate, to support your hypothesis, you need to collect the right kind of data in the right setting. So I guess we'll switch over to an actual experiment here. This is one that I had the headline for this article, the title on the earlier slide. They did a manipulation where they cut some leaves and they were observing how that influenced the processing by this herbivore. So kind of an interesting plant insect relationship. This insect cuts the leaf, rolls it up. I don't remember, lays eggs in there maybe. And they were suspecting that maybe these leaf shape cues are important for determining whether the insect is going to see that as a viable leaf. So if this is what you're going to eat or where you're going to lay your eggs, you got to choose the right starting point, something that works with your biology. So a couple different kinds of leaves. And over here we've got some of their data. Left is lobed and right is non-lobed. So I think these are artificially cut to be lobed. And they compared how that attracted insects. So they had the same starting plant species. So it's the same nutrient content, smells the same, looks the same texture, same shade of green. And they just manipulated the shape, by cutting it. And they're reporting their data here, proportion of leaves used per stem. And you can hopefully see we've got these bars shown here. So bars with error bars. What does that mean? What is an error bar? Basically, yeah, range of results, range of variation. So in biology, things are not identical across individuals. I'll just maybe do an example using height. So you could have lots of different individuals that are all growing up to a certain point, and you got minimum growth, maximum growth. So somewhere in there is the what often gets reported, the mean or the average. And I won't get into the math of it. But it's a visual depiction of variation. So if all the measurements are pretty close to the mean, you get a small error bar. If all the measurements are very widely distributed, some very short, some very tall, you'd still get a similar mean, but a much higher variation around the mean. So these are relatively small, I would say. One way to evaluate if these things are different, that's one of our comparisons. Just visually, you can kind of look at those and see if the error bars of the right-hand thing, those overlap with the error bars of the left-hand measurements, then that's probably something that's not all that different. All right, so are they different or not? So we have proportion of leaves used per stem. What is the null hypothesis in this experiment? They cut the leaves, they didn't cut the leaves. What's the null hypothesis for the effect of cutting leaves? No difference? Yeah, right. So the null hypothesis, they're doing this manipulation. Again, they sort of want to find that leaf shape is important, so they change the leaf shape and they're seeing if there's a difference. But the null hypothesis, the fallback, is that there's really no difference, that they didn't change the behavior of the insects as a result of cut. And there's a value that you hopefully have seen a few times in your biology career by now, called the p-value. And the p-value is basically an estimate of the chance that the two measurements could in fact be the same. So taking into account natural variation says that the lobed leaves and the non-lobed leaves could be the same. This gives it about a 38% chance that that could happen. All right, so p-value equals a few different ways you can write these. I'm going to go ahead and say it this way. So a numerical measurement of how likely it is that the null hypothesis could be true. So in this example, null hypothesis that those two measurements are not different from each other, that could be true. And what I had on that slide is the threshold. The usual value is a p-value less than 0.05. So frequently this cutoff of 0.05, if the p-value is that low or lower, people take that as what we call a significant difference. Something students like to put on papers, they say we got a p-value that was low. That meant that our results were significant. Sort of. I think the meaning of significant in the common language is different from the scientific meaning. So a difference that is significant, this is a meaning that's rather specific in science. So significant relates to this low p-value. So a significant difference or a significant correlation or something like that. Significant meaning that it's more than just would happen by chance. So 0.05 is just 1 in 20. But the thinking is that if you set your experiment up correctly, then this should be enough to support that there is a real difference. So on the left side here, this comparison is not a very low p-value. And as we can see visually, they're not all that different from each other. How about over here, lobed, lower than non-lobed? Now we have a p-value 5.9 times 10 to the minus 4, 0.0005. That's a lot lower than 0.05. And that says that cutting by the insects happens significantly more in leaves that are not lobed. And over here, also low, significantly different chance that the leaves get rolled up in the non-lobed leaves. So you might have seen, I don't know, I think it's, if you're not familiar with the p-value cutoff, you read these papers, anything that they want to draw a meaningful conclusion from and make a powerful statement about at the end, say that cutting these lobes in the leaves significantly changed feeding behavior, they need this kind of evidence to support that. All right, still doing okay? All right, so this is a significant difference. There are other more complicated variations on that, but just basically a nice, easy way. You're measuring one trait. They've managed to quantify the amount of cutting and the amount of rolling. Another type of evidence is a correlation, where you compare two things and you want to see if one is correlated with the other. And they might not tell you specifically that one causes the other, but it's a good starting point about two things that are related, functionally related, or maybe related to a third thing. So here the alternative hypothesis is that these things are correlated. That wood that is more red is going to have more phenolics in it. Right? So what is the null hypothesis in this kind of arrangement? Draw this out for us. that they're not correlated, right? So the null hypothesis is that they're not correlated. What does that mean? Basically means, I'll draw a straight line there. So the null hypothesis here is basically that the line that you would draw to connect these points, the best fitting line, is not any different than if you just drew a horizontal line. And what that means, a horizontal line means that if you change the value of x, that has no change, no discernible change on the value of y. All right, let's see. So our null hypothesis, red wood will not have more or less phenolics than more lightly colored wood. Great. So basically no relationship between the two things on the axes. So these look like the lines are a pretty good fit for the points. You can see that natural variation on either side of the line. And from one of the p-value, again, the p-value evaluates the chance that this fit is, I guess the p-value will tell you how well the zero slope line fits. I think that's an okay way to say that. I don't teach a statistics course, but I brush with statistics once in a while. Another feature you see often when they compare values with this correlation test is the R-squared value. And R-squared heading towards 1, values closer to 1 mean these points fall very close to the line. R-squared closer to 0 means that they're not really a good fit for that line. Okay, so we talked about the cutoff for the p-value, the R-squared value. I think people usually report a p-value also when they do R-squared, but in case you encounter this number, it's something means that line fits the points very well. Closer to 0 would mean that it hardly fits that line. And I guess just sort of a personal side note, statistics aside, I feel like anything that's worth reporting, taking home and taking as truth, you should be able to see it visually on a graph like this. So if somebody tells you that phenolics are correlated with the color of the wood, they show you this, I'm pretty well convinced by that. This looks like way more than random that the points increase on the x as they increase on the y. You don't always see them this clear. I guess this one on the far right is becoming a little worrisome. A lot of times people have data where they draw one of these lines and the points don't really look like they fit the line very well. I find those a little harder to believe. Okay, so a little bit more about experimental design. We started off talking about what happens if I fertilize a plant. To get statistical power, as they say, you got to observe the same difference multiple times, right? So if I take two plants and I water one and I don't water the other, and one grows better, I still haven't ruled out a lot of what could have happened by chance. But if I take 30 plants and I water those and I randomly put them in different places in the growing space, and I take 30 plants and I don't fertilize, I say water, water some, don't, sorry, say it again. Don't fertilize some, fertilize others, take 30 of each, randomize their locations. If you still see that difference across this large number of individuals, that contributes to the p-value. So like here, we've got lots of points. You could draw a line with 2 points. It wouldn't be a very convincing line. On here, it looks like they put their proportion of feeding into categories. So there's some numbers at 1, so 100% leaves used, 3/4 of the leaves used, half of the leaves used, and so on. But each of these rows across has lots of points on it. So I don't know how many they had offhand, but lots of individual plants were used for this experiment, lots of points on both of these. So I guess I can't really answer for everything with this, how much is enough answer to that question. For, I see 30 seems to be a pretty popular number, kind of a sweet spot of affordability and statistical power. This is a set of plants that are growing up for a population genetics study done by somebody at UW-Milwaukee. Does some really cool stuff. He's got a population of plants that each has their own genetic signature. You can find out when they pollinate each other, they can find out who the parent was every time. All right, I think we probably asked that question already. So let's say we go out into the world and you want to isolate an effect. You might be faced with having to have more replication in these instances. So we talked earlier about controls. This is a nice little example of controls. So this is plants growing in the field, and they manipulated the amount of sand that stuck to them. So they found out that there was a plant that typically had a lot of sand on it, and they were running a hypothesis that this would prevent animals from chewing on the plant. So a nice way to do controls is to manipulate everybody. So all of these were manipulated. They washed off the sand, and then they replaced sand that was colored, or sand that was green colored, so they could control for a variety of different effects. Did the sand physically prevent chewing? Did it visually signal that these were not a good thing to eat? And they could separate out those possibilities. So they had plants without any sand, plants with tan sand, and plants with green sand. And I have my p-values on here, but what they found on the y-axis here, proportion of leaves chewed. And again, hopefully you can see from the bars and the error bars, the sand removal had significantly more chewing than either brown sand added or green sand added. And these two were not significantly different from each other. So they demonstrated that the sand prevents chewing, but it might not have a big role in keeping the herbivores from getting there in the first place, right? So if they see a plant with green sand on it, it might look like one that doesn't have any sand on it, but still prevents them from chewing on there. So this one, in one experiment, you could do presence of sand and color of sand and see which one of these has the biggest effect, if any of those effects are different from others. We make sure you can collect data that are quantitative. So best kind of data to run a statistical test on are data that you can put into numbers. So number of organs, mass of organs, surface area that gets chewed. So one more time back to here. For this study, they did a visual observation category breakdown of no damage, 1/4 damage, 50% damage. So even doing it that way allowed them to do a numeric analysis on the data. All right, so hopefully you agree there are a lot of advantages to working with plants. Anybody sold on experimenting with plants yet? Maybe? What advantages do plants have over animals? Easier to keep alive for some people. Yeah, at least I don't envy my colleagues who have to come in and feed their fish every day, frogs or whatever's getting. Grow and develop quickly, some of them certainly. Fewer ethical questions. Honestly, that's one of my favorite parts. So if you do a science experiment, you have a research project here and you're doing it on any kind of animal, you've got to get special approval. They say, what are you going to do to make sure that that animal is taken care of humanely? If you do end up killing the animal, kill the animal humanely. People don't really ask those questions with plants. So you're going to dispose of them, right? Yeah, so I guess a little less maintenance. Can grow quite a few of them in small spaces a lot of times. They're a little bit simpler. I mean, you can put them in one place and keep them there. So whatever you put in their pot becomes their food. So that's maybe not too different from some animals. I don't have to convince you to work on plants. This part of the presentation, I just wanted to kind of highlight the different levels of control that you might have over the organisms you're working with. And the first level here is where you can basically control everything about their environment. So you can grow plants just like you could, say, bacteria or yeast colonies or something. Grow them in a completely sterile environment where they are the only cells in that environment. So not every plant deals with this well, but there are some. This is a fern gametophyte. And it's being grown in a sterile environment, doesn't have any other organisms, and also a chemically specific environment. So they found out the nutrients that plants need, and you can make up a chemical mix of just what they need to grow. So if you wanted to test if you alter any of these ingredients, you could change one, leave everything else the same, and get really strong evidence for the importance of that one molecule. So this is from a study where they combined the fern gametophytes with a type of bacteria. They found that it would change the sex determination, the amount of male and female sperm-producing cells and egg-producing cells that are on these gametophytes. So very specific controlled environment. Controlled environments in the greenhouse can control the nutrients, the light, the water, temperature. But a lot of features, particularly plants, a lot of plants are not easy to cultivate, or they might have, they take a long time to grow up, or they don't flower under the conditions you have them on. They might be rare plants in nature. So running manipulations of flowers in nature is a little different from taking those plants out and trying to grow them in the lab. I guess the advantage here, another advantage is that their populations are already present. You don't have to establish anything. You just locate the plants and they will be flowering at the time when they flower. So example here where they had a treatment where they removed the corona. So this is a close relative of daffodils. And this little cup here, it's called a corona. Not a good year to say corona, huh? So corona, just a crown, or it's basically like a bit of the flower in between the petals and the next organs in. And they went in and they experimentally took that out. So manipulating flowers, another thing you can do with plants pretty easily. You can just cut away organs, see what happens. So here we have present, so left intact in gray, and then the ones with the corona removed. And this was showing different kinds of flowers with different relationships of their reproductive organs, and made a big difference in these S flowers, much lower seed set if the corona was absent, across a couple different treatments here. Okay, so field manipulations. Here's a field manipulation. They did a lot of manipulation, but they still did it on plants growing in the field. So that would take advantage of their natural habitats where they're already fit to grow. They're already in the environment where the animals would be visiting them to eat them. Pretty popular way to manipulate plants in the field is to see how their reproduction is affected. This was a test to see whether this species could fertilize itself. And the light-colored circles at the bottom are flowers that they emasculated. They took the stamens off of those flowers, prevent them from, they'd be physically unable to fertilize themselves because they didn't have any pollen. And in those cases, their seed set was way down, basically indicating that they weren't getting a lot of pollen from other flowers. So taken to them by pollinators, looks like this species relies on self-pollination for a lot of its seed production. So again, hypothesis, I think this thing self-pollinates a lot. I'm going to go in and remove its ability to self-pollinate and see if the pollen that is delivered from other flowers is enough, and definitely a big difference between those amounts. So still on manipulations here. Also possible to look at natural populations and see, look for patterns. So this involves simply observing what's already out there. And this was a study looking at the relationship between temperature and other environmental factors in a group of plants that has variation in their leaves as far as how how deeply they're divided. These different species growing in South Africa, do they have different climatic tolerances depending on how their leaves are put together? I guess another way to look at that is to ask the question, is climate responsible for the differences we see in the leaves? So they've evolved different leaf forms. Why is that? Their hypothesis was this is an adaptation for different temperatures and other climate factors. This one, not so much looking at the form of the plants, but instead looking at their genetic differences. So getting at the DNA of the plants, and they broke them down into these groups that are geographically separated, and turns out that they are also genetically different. So this tells us that they're probably not exchanging genetic information across those boundaries. So the ones in the green zones are pollinating with each other. The ones in the blue zones are pollinating with each other, but it looks like there's not really pollination between blue and green or any other pair of populations. Sometimes you get natural variation that allows you to do what we would call sometimes a natural experiment. This means basically when you have an experiment, you intend to have a difference, right? You set your control and your treatment. A natural experiment is where a difference exists and you sort of capitalize on that to see if that difference has the effect that you think it might. So this is a group of plants that have very long floral tubes. So there would be nectar at the base of this flower. And a lot of times this shape of flower corresponds to an animal that's able to get the nectar, right? So somebody's able to get the nectar that's way at the bottom of this tube, an animal that'd be able to get that effectively, efficiently, because it's not a lot of food, but it would be enough for somebody who can get that repeatedly from different flowers. And this one tells us that the combination of parents, that there's like a preference for pollen from short flowers to be received by short flowers with ovules. All right, let me spell this out a little more clearly. All right, so seed set, a good measure of performance. If you take the ovules produced by a plant with short flowers, they produce more seed if they are pollinated by a pollen grain that comes from a short flower. So short by short produces a lot of seeds, and then over here, long by long. So long female parent, long male parent, that combination also produces a lot of seeds. But long by short is not a lot of seeds. Short by long is not a lot of seeds, and anything intermediate also low amount of seeds. So this is like a reproductive isolation that may well drive these two different sizes of flowers to become different species. Another one here, this is an underwater aquatic plant called Utricularia or bladderwort. We have some around here. They just went out and collected the traps of this carnivorous plant, and algae was in there, and they could correlate that with the growth of the plant. So it's one that we think of as carnivorous, of trapping animals, and they demonstrated that sort of accidentally, maybe intentionally, also capturing algae was a way this plant could get some nutrients. All right, so I realize I got a lot of these. This is our last one. Another type of study you might see more on a bigger evolutionary scale is maybe tracking how adaptive certain kinds of evolutionary change are. So this is a study across a whole bunch of monocots, so grasses and relatives, lilies, asparagus, and checking out the types of underground storage organs they have. So bulbs are modified leaves, corms are modified stems, rhizome is a different kind of modified stem, tubers, and they got these coefficients for how they transition. So transitioning from a corm to a rhizome is more popular than corm to tuber and so on. So by having lots of different changes happen over this big tree of life, they can develop some patterns and say, maybe this is developmentally connected. Maybe it's easier developmentally to go from one of these kinds to another. Kind of setting the stage for future research questions. All right, so I guess we have, I mentioned the research project. This is a good way to hopefully get familiar with the kinds of papers you might be reading related to that. Feel free to ask me questions, email, in person. If you are thinking about a research topic, I don't think of the proposal as like the end product. Usually it's the start of a conversation, and then we can narrow you down and help you find your papers that are interesting for you. All right. Well, if we don't have questions, I think that's probably a good stopping point. Okay. For the near future, I think we'll just shift what's scheduled to happen by one class period. And I'll let you know as we go.